A warm welcome to all the learners to my channel Microbiology Tutorials. Today I am going to discuss a relatively new topic which is called as omics. Omics is a new approach to life science, and it have revolutionized the the basic science in which we deal or we take a lot of aspect together. If I explain you in detail, we know that life science is an ever-changing field, which means that there is a new innovation, new technology, new inventions, then new diagnosis are coming, and a lot of innovations in the last 40, 50 years have been introduced. Moreover, in the biological sciences, we know that there are different parts or different aspects in biology. It include systematic biology, or we can say structural biology, molecular biology, ecology, environmental biology. So if we see that earlier or even today also, when we deal with these topics, so for each and every aspect, we have to approach it independently. Now, the if we think like that if we bring all these aspects together on one platform and we deal with it at combining all these parts together, it will certainly help to find the solution of a problem more robust, in a more robust manner, more precise, it would be more precise, and we will get a very cutting edge solution for all these problems. So it have almost revolutionized the thinking regarding our basic sciences. Now, if we think more in detail, we can say that different aspects of the sciences is have been brought on one platform, and instead of dealing all the subjects independently, we have aggregated all these things together, and we try to find the solution or at a more molecular level, we can say, so that we can get the result more precisely. So there is a paradigm shift in the approach towards the basic science, where on one side we used to deal with any problem either on a genetic level, or we may say any on the protein level or a single compound level. So here all these different parts or different aspects or different branches have been together combined, and we used to see all those parts on a broader scale, and we can easily find the solution to any particular problem persisting to that condition. Either it is medical sciences, or we can say agricultural sciences. So usually, so these parts have been combined together, and we utilize all these technologies together to find the real root cause of any problem. So today we will see how the omics have totally changed the life science field. So in biology, the suffix omics refers to a huge biological molecules. The broad analysis of large biological molecules was needed to be studied in detail as a conclusion of Human Genome Project in 2001. The Human Genome Project revealed that the human genome contained lesser number of genes and biological processes were regulated not particularly on DNA sequence, but involved various other processes. And with this evolved the new branch of study termed as the omics, given by Hood and Robin in the year 2013. Now, if I give you a brief explanation about the Human Genome Project, so it was started in the year October 1990 and it ended in the year April 2003. So it was a 13-year period in which there was a consortium of 20 research centers and the scientists from different countries have participated to study the entire human genome structure so that they could find out the the characteristics which is present in the humans at the genetic level. And they were motivated to study the Human Genome Project by the landmark discovery of the DNA, which was discovered by Watson and Crick in 1953, and further more studies further made interesting to for made scientists to study these field. And then came the era when the recombinant DNA technology came into it came into existence. Then Sanger sequences, which was developed in the year 1977, and further by the development of polymerase chain reaction by Carey Mullis in the year 1983. So all these things greatly revolutionized or made this field a great impact on the study of genetics. So scientists thought that if we could able to study the entire genomic structure, it would be easier to understand different type of diseases and to find the cure for it. So then the need for after the completion of Human Genome Project, the need for omics arose. So what is omics? We will see. The technology deciphered the cell, tissue, and organism in a holistic way around central dogma for the detection of genes, mRNA, which was called genomics, mRNA, transcriptomics, proteins, proteomics, and metabolites, that is the metabolomics in the samples, a specified biological component. Since the technology is non-biased, they are also referred to as high-dimensional biology, where the integration of these in these systems biology, following the discovery of DNA structure by Watson and Crick in 1953, a series of inventions and discoveries followed. The development of PCR by Carey Mullis opened all possible channels in molecular biology research. So omics was the it arose from the molecular biology science. And further, more research or more work was done to develop a completely new field. So we will see here what are the highlights of the omics science. And further, since this genomic structure, study of genomes, as it is a human genome has very large, consist of many billion base pairs. It is made up of billion base pairs. And to study each and every aspect of the structure, we have to maintain their data. And to maintain the accurate data manually is quite a cumbersome and difficult work. So with that, the need for the use of computers and software was felt. And with this, the when in the biological science research, the system computer or software system as it was introduced, so the use of these also became mandatory. The use of computers became mandatory because with its use we can it helps to maintain the many different types of data structures related to the proteins or genes quite efficiently and effectively for a long duration of time. And this gave birth to a new biological science, that is the bioinformatics. So the area of bioinformatics incorporate a wide range of biotechnological subdisciplines that are highlighted in by both scientific ethics based on biological sciences and deep knowledge of computer science and information technology. So it is an integration of biological sciences with with computational technology for its use in the life science. So some of the examples of many fields in bioinformatics include computational biology, the uses of data-based solution to the issues in bioinformatics. Genetics, it is a study of heredity and the gene diversity of inherited characteristics or features. Genomics, that is the branch of biomolecular biology that works in the area of structure, function, evolution, and mapping of genomes. Proteomics, the study of proteomes and their features. Metagenomics, the study of genetics from the environmental and living beings and sample. So usually these 4 5 are the some of the important parts of the omics. In addition to these, we also include the, transcriptomics. It is the study of complete RNA and DNA transcriptase. Phylogenetics, the study of relationship between groups of animals and humans. Then metabolomics, the study of biochemistry of metabolism and metabolites in living beings. System biology, mathematical designing, analysis, and visualization of large sets of biodata. Structural analysis modeling that determines the effects of physical loads on physical structures. Molecular modeling, the designing and defining of molecular structures by way of computational chemistry. And pathway analysis is software description that defines related proteins in the metabolism of the body. So here you can see that study on the basis of DNA, RNA, protein, as well as the use of software, then which is used for designing the molecular models. We can say the molecular docking part is included in this. So all these parts were included, and a great advancement in the technology from the technological point of view was made in this field, and it gave great benefits in studying the the pathway of different type of diseases or their characteristics, as well as also the the basic functions of the cells, and how they how they appear, or how they functions, normal functions, or how their characteristics changes under any type of abnormalities. So all those, if we include all these branches, we can easily understand the different aspects of the of the biology. So here, if you can see here, so basically you can see here that this is the DNA where the information is stored in genes and other DNA sequences. So genes contain recipes for proteins, and this is the protein machine. So here, this is the pathway through which the entire function or entire synthesis of protein from the genes, DNA to genes and genes to protein takes place. And in the entire this entire pathway is studied under the field of omics. And within these parts, there we not only this central we go to the central dogma. Basically, the central dogma is the backbone of the molecular biology science, and it includes all those parts through the entire structure from the genetic makeup to the formation of protein. So this is a quite long step, or we can say complex process, through which the entire things or entire genetic makeup goes, or it is processed to form a protein structures, and how these protein structures develop from its primary structure to quaternary structure, or we can say functional structure. So all these things are to be studied through this technology, or we can say through this branch, and how they modify themselves and adopt themselves to different conditions are included in this branch. So the central dogma, we can say, is the mainstay of this science. So, omics is the modern scientific discipline that employ bioinformatics, system biology, bioanalytic techniques, next generation sequencing to investigate the complete set of biological molecules within an organism, ranging from genes to metabolite. The term omics is derived from the Greek suffix om, meaning all constituents of a system, means the cell, then metabolic pathway, nucleus, gene, DNA. So all systems are included. All the constituent, they include all the constituents of a system, reflecting its systems level approach to studying cellular and organismal functions. Omics deals with various branches such as genomics, transcriptomics, proteomics, and metabolomics, each focused on a different class of biomolecules, that is DNA, RNA, proteins, and metabolites respectively. Unlike traditional reductionist methods that analyze one gene or molecule at a time, omics allows researchers to examine these components collectively, providing a holistic view of biological process of an organism. This system level analysis is crucial for understanding the complex interactions underlying health and diseases. Many clinical conditions such as cancer, diabetes, cardiovascular, neurological, and immune-related disorders are multifactorial, arising from a combination of genetic, sedentary lifestyle, environmental, and biochemical influences. By analyzing large-scale biological data sets, omics helps unravel the molecular mechanism that drive such diseases. Now, here one thing I'd like to highlight, that these diseases are not only caused on the genetic level only. So there are some environmental factors, or some other factors which are also responsible, and which fact and to certain conditions, which are the important factors which are responsible, can be easily studied through this technology. So genomics, if we go in detail, genomics is the new science that deals with the discovery and noting of all the sequences in the entire genome of a particular organism. So the genome can be defined as the complete set of genes inside a cell. Genomics is therefore the study of the genetic makeup of organisms. Genomics is an entry point for looking at other omics science. So it is a gateway. First of all, obviously, we have to study the genetic makeup of any individual or any organism to understand the entire structure, or the entire activity of any cell or tissues. So it is we can say that it is the entry point for the omic science, and the information in the genes of an organism, its genotype, is largely responsible for the final physical makeup of the organism, referred to as phenotype. However, the environment also has some influence on the phenotype. So as earlier I told that Human Genome Project played a major role in development of this new science. DNA in the genome is only one aspect of the complex mechanism that keeps an organism running. So decoding the DNA is one step towards understanding the process. However, by itself, it does not specify everything that happens within the organism. The basic flow of genetic information in a cell is as follows. The DNA is transcribed or copied into a form known as RNA. The complete set of, and this first of all, DNA to DNA is formed. The process is called as replication, and DNA to RNA, that is called as the transcription. And that is the science that from that only the derived the term that is the transcriptomics. And the complete set of RNA, also known as transcriptome, is subject to some editing, cutting and pasting to become messenger RNA, which carries information to the ribosome, the protein factory of the cell, which then translates the message into protein, which constitutes the field that is the proteomics. So what is the proteomics? So proteins are responsible for an endless number of tasks within the cells. The complete set of proteins in a cell can be referred to as its proteome, and the study of protein structure and function and what every protein in the cell is doing is known as proteomics. The proteome is highly dynamic and it changes from time to time in response to different environmental stimuli. The goal of proteomics is to understand how the structure and function of proteins allow them to do what they do and what they interact with and how they contribute to life processes. An application of proteomics is known as protein expression profiling, where proteins are identified at a certain time in an organism as a result of the expression to a stimulus. Proteomics can also be used to develop protein network map, where interaction among proteins can be determined for a particular living system. So here it's very important in the formation of drug discovery also, where the response of any drug or any protein, or the effect of the drug on any protein metabolite can be easily studied. Next, coming to the metabolomics. So metabolomics is one of the newest omics science. The metabolome refers to the complete set of low molecular weight compounds in a sample. These compounds are the substrates and byproducts of enzymatic reactions and have a direct effect on the phenotype of the cell. Thus, metabolomics aims at determining a sample's profile of these compounds at a specified time under specified environmental conditions. Usually, these metabolites are usually low molecular weight products, and it is easier to quantify them or to study them and to interpret the result easily. So genomics and proteomics have provided extensive information regarding the genotype but convey limited information about phenotype. Low molecular weight compounds are the closest link to the phenotype. It helps in the study of phenotypic structures or changes in the phenotypes. So you can see here that when we deal with the DNA, it is the genomics level. On the RNA, it is transcriptomics. Proteins, it is proteomics, and metabolites, it is metabolomics. So if we keep in mind that omics research uses multidisciplinary approach requiring integration of molecular biology, biochemistry, computational biology, bioinformatics, statistics, and clinical sciences for data generation, storage, and interpretation of massive biological data sets. Recent advancements in high throughput technologies, artificial intelligence, and machine learning have enabled the development of multi-omics approaches where different omics disciplines, genomics, transcriptomics, and proteomics are integrated to gain deeper insights into complex biological systems. Such integration facilitates the mapping of the entire genome, transcriptome, proteome, and metabolome within a short time frame, offering a molecular and biochemical snapshot of an organism. Next generation sequencing plays a pivotal role in contemporary omics studies, enabling rapid and cost effective sequencing of DNA and RNA. This has revolutionized biomedical research, allowing for the discovery of novel biomarkers, understanding disease pathophysiology, and identifying potential therapeutic targets. Omics technologies are widely applied in diverse fields, including biochemistry, pharmacology, clinical diagnostics, and personalized medicine. The primary objective of omics research is to collect, integrate, and analyze comprehensive biological data to address contemporary challenges in human health, agriculture, and in the pharmacy, and also in environmental sciences, pharmaceutical sciences and environmental sciences. The knowledge gained from omics-driven studies is instrumental in advancing precision medicine, developing targeted therapies, and improving disease diagnosis and prevention. Why are omics technologies important? Omics, as we have already discussed, the importance of omics and how it can be identified for different genotypic and phenotypic structures. So, when we are studying about the genomics or genetic, we can only uh, describe any condition or any disorder at the genetic level. But with the introduction of omics and with the metabolomics, we can also characterize any of the conditions at the phenotypic level also. Means the appearance, based on its appearance. So why are omics technology important? Omics sciences have transformed biological research by offering the fundamental architecture and dynamic functions of living systems. By deciphering the interplay between genes, proteins, and metabolites, researchers can better understand how cells operate and adapt over time. Beyond basic sciences, omics technologies are pivotal in applied research. In healthcare, they support the development of personalized therapies as tailoring cancer treatment based on an individual's genetic profile. In agriculture, too, omics tools are used to develop climate resilient crops, enhance food security. These applications demonstrate that omics technologies are not only advancing our understanding of life at the molecular level, but also shaping the future of biomedicine, biotechnology, and sustainable development. Now, here, from its introduction to the current scenario, there have also been a great change in the omics. So we will see what till now what have there have been evolved from its uh, uh, previous version to the current version. How much the field of omics have changed over the years. Advances in biological techniques and computational tools have given rise to a transformative new field of science, omics, the edge methods to study biological. Over the years, advances in biological techniques have transformative new field of science. The interdisciplinary domain integrate cutting systems at scale. So we see here that earlier we used to study on the genomics level, then came proteomics, then metabolomics, then we added the technology of computer softwares, then computer aided design. Now with in this artificial intelligence era, we are using to improve the technology of omics through AI or through machine learning. So it have still there have been always been a scope of improvement. We will see in the coming slide that more and more branches is getting added to the field of omics. So these improvements have made omics more useful, allowing us to learn more about how molecules work in living things and speeding up discoveries in medicine, agriculture, and biotechnology. So it is not only one part. It is not only it is able to interpret result on basis of healthcare or medicine, but it is also contributing in the drug discovery, then in the field of agriculture for better crop yielding, and also with the use of the computer softwares or to use of technology, they are making some one of the cutting edge technology. So it is a fusion of technology and biological sciences for betterment of the human life. So the it have greatly changed our thinking about the medical science. So omics technology also referred to as high dimensional biology encompasses the cells, tissues, and organisms in a manner that integrates the data from various platforms and helps in interpretation. It primarily detects the genes, mRNAs, proteins, and proteins and metabolites in a non-targeted and non-biased manner. The integration and interrelationships between networks of biological processes termed as systems biology. The approach provides hope for unraveling the intricate details in various aspects of biology and accelerates innovations in healthcare. Understanding the various dimensions encompassing not only the three levels constitute the central dogma of life, but also the intermediate metabolites is significant for the scientists to cover new horizons in drug discovery and disease regulation. Omics technology applied majorly for accurate understanding of normal physiological process and gaining knowledge related to disease process, which involves screening, diagnosis, and prognosis that provides an understanding of the etiology of diseases. The progress in omics started from the development of genomics, further followed by transcriptomics, and finally the proteomics. And the term was coined in 1994 by Mark Wilkins. This was possible because advanced development in techniques like high resolution two-dimensional electrophoresis, the cascade of events in omics is depicted in the figure. And the advantage of the omics study is that they reveal specific results that promote understanding. As the omics technology is of immense potential, they have been explored in various branches of medical and health sciences. This technology can help us understand the etiology of disease condition through the process of screening, diagnosis, and prognosis, and also for the biomarker discovery to be made easy as they involve simultaneous investigation of multiple molecules. Further, omics is of great use in drug discovery and toxicity assessment. Pharmacogenomics deals with the connection of genomics and pharmacology to examine the role of inheritance in individual variations in drug response utilized into individual and optimize drug therapy. They help in the field of oncology to evaluate rigorous systemic toxicity and unpredictable efficacies that are hallmarks of cancer therapy. So here, if I show you more, so here you can see now here genomics was there. We discussed transcriptomics, proteomics, and metabolomics. So now epigenomics. So it is a histone modification, DNA methylation, 3D organization of chromatin. Then cellomics, cell functions, phenotypes, morphology, biochemical activity, etc. And single cellomics. So more and more branches are being added to increase the efficiency and precision of this cutting edge technology of omics. So this is the glossary of some terms. So the chromosome, which is a group of coiled strands of DNA containing many genes. DNA, a molecule found in cells or of organism that encodes genetic information. Gene, a biological unit that codes for distinct traits or characteristics. Genome, the complete set of genes in a cell. Genotype, the genetic constitutions of an organism. Metabolome, a complete set of low molecular weight compounds in a cell at a given time. Phenotype, the physical appearance or observable characteristics of an organism. Proteome, complete set of proteins in a cell at a given time. RNA, a molecule derived from DNA by transcription that either carries information, provides subcellular structure, transports amino acids to or facilitates the biochemical modification of itself or other RNA molecules. So this is the a brief lecture on the omics. It is a diverse field, and as you have already seen, as I discussed here, that multiple branches are included. So have tried to give you a brief idea about this new technology, and which could transform, which have the capacity to transform the entire biological sciences, and can also prove important landmark in the discovery of some of the uncurable or fatal diseases, and we can save the humankind, human being from some of the very serious, or we can say that fatal diseases to which till now uh, much even more research are needed. So with the introduction of omics, it could be easier for the researcher to find solutions for more and more issues or diseases which are still a challenge for the humankind. With this, I conclude my today's lecture. I hope you have certainly gained some, you certainly gained from my lecture, and please do subscribe and like my channel so that I get motivated to bring more information in the field of biological sciences. Thank you.